Hi, my name is Farrell, and today I'll be talking to you about experiment design. This is part of our series on biology, so if you haven't looked at the other videos, be sure to check them out. If you have, let's dive right in. Experiments. We all know what experiments are, and they're useful because they allow scientists to measure the effects of a certain change in a controlled environment. And it's important to remember that experiments aren't the only method of scientific inquiry. Other examples include observational science or historical science, where we look at data that's been collected over the past year or 10 years or 100 years even. These are all methods of scientific inquiry, but experiments are a powerful tool for scientists, so it's important to know how to make good experiments and get good results with those experiments. Here's an example of a very famous experiment, very important. You won't need to know how it works exactly just yet. This is an example of a very important scientific experiment. You won't need to know how this works for now, but just know that this is the Hershey-Chase experiment. And what this experiment did is that it demonstrated that DNA was the genetic material as opposed to proteins. And so you can see how important experimentation is for scientific advancement. So the first part of making an experiment is formulating your hypothesis. And your hypothesis should be concise and testable. It would usually outline a correlational relationship. So we usually phrase that in like an if-then statement. Like, for example, if I give patient drug A, then they'll be cured of cancer, or something like that. My example hypothesis here is greyhounds run faster than poodles. And that's a good hypothesis. It's concise and it's testable. This is something we can test in a lab. And so an example of a bad hypothesis would be this monstrosity here, which is not concise at all. And yeah, we could make that better. This one at the bottom, greyhounds are better than poodles. That's also a bad hypothesis because it's not testable. That's not something you can test in a lab. Don't do this. Do something like this. Greyhounds are faster than poodles. Concise, testable, correlational relationship. When we design experiments, we would want our results to be both accurate and precise. So what does that mean? Well, if we look at this graphic here, we can see that this target right here, somebody's shooting at this target and they're both imprecise and inaccurate. So what would that mean for our experiment? Well, this would mean that our results are very spread out and it doesn't hit the target, meaning it doesn't correlate to reality well. This one right here, this one is not accurate, but it is precise. In terms of an experiment, that would mean not very spread out data, so your data is all very compact, but they don't reflect what's true in reality. This one is imprecise, but accurate. So that means that the data reflects what it is in reality, but it's very spread out, so that's not very good either. And so the best case scenario is having data that's both accurate and precise. Data with low variance would mean that it's both accurate and precise. So when we talk about experiments, we're going to have variables in these experiments. Usually we have three kinds of variables: controlled variables, independent variables, and dependent variables. The controlled variables are going to be things that you keep the same throughout the experiment. What does that mean? We're going to talk about some examples later. Independent variables are variables that are manipulated by the researcher, so things that are changed by the researcher in the experiment. And the dependent variables are going to be the variables that change according to the independent variables. So when you change the independent variable, the dependent variable changes. So this is a very basic experiment. You have two plants. One of them receives nothing, and the other one receives salt. So what's going to be the effect on the plant height? You could try to identify the control, independent, and dependent variables here. You could pause the video right now if you wanted to try it. Controlled variables are going to be ones that stay the same for both these groups. Usually everything except the independent and dependent variables are going to be controlled, meaning we'll have to make sure that they stay the same. An example would be the type of plant that I use, or the type of soil that I use, or the amount of sunlight that I give them, or the amount of water that I give them. Anything you can think of that isn't the independent or dependent variables, those have to be controlled, ideally. And so the independent variable was different between these two. Well, we're adding salt to this one, so that's going to be our independent variable, whether or not there's salt. And the dependent variable, it's in the title right there, effect of salt on plant height. So we're going to measure the plant's height in response to this salt. So the height depends on the independent variable, which is the presence of salt. And so our height is our dependent variable. When we're doing experiments, usually we'd have a couple different groups. Control group is going to act as a baseline. You could think of this as the default. Group 1 is clearly the control group because it has nothing added to it, and it's served as something that we can compare against. So it's basically acting as a baseline. And so the experimental group is the group that you do things to. So in this one, we're adding the salt to the experimental group. Group 2 is our experimental group. So positive and negative controls. This is a little confusing the first time you encounter it, but it's actually not that hard once you start to understand it. So a negative control, this one's easier to understand, is the group that receives no treatment. In medical trials, usually the group that receives no treatment, i.e. the negative control group, it will receive placebos. So they'll think that they'll be receiving the treatment, but they won't be. Basically, we want to keep everything the same except for the independent variable. Like for example, this is going to be a negative control. So we keep everything the same. We keep the soil, the type of plant, the amount of sunlight, everything's going to be the same except for the independent variable. A positive control is a group receiving a treatment with a known result. So this is basically, we're giving them something that we can compare it to. So in this example, we have three groups of crops here. And in our first group, there's gonna be no fertilizer. And in our second group, there's gonna be fertilizer A. But we already know that fertilizer A has an effect on plants, and we know the effect that it has on these plants. And so that means we can compare it to fertilizer B. And so fertilizer B here, this experiment is meant to test the effect of fertilizer B on these crops. So since we have fertilizer B, we can compare it to both group 1 and group 2. And so group 1 is our negative control because everything stays the same except for the presence of fertilizer B in the experimental group. This one doesn't have fertilizer B, but this one has fertilizer A, and we know the effect that fertilizer A has on plants. And so that's something we can compare it to with fertilizer B. And so group 2 is the positive control, group 1 is a negative control, and group 3 is the experimental group. When we do experiments, we want to find methods of reducing the variance in the data. And so the variance in the data basically means how accurate and precise it is. Something we can do is increase the sample size. So if we look at our first example here, we're just using one plant in each group, but we could increase that to 5 plants or 10 plants or 100 plants. When we do that, our data is more reliable because there's less things that are just left up to random chance. Or we could do more trials. We could perform the experiment several times and take the average. And that would also reduce the amount of variance because you're getting more data. You can average it out. One last thing you can do is eliminate confounding variables. And confounding variables basically are variables that are supposed to be controlled, but they're not. And they could be affecting the outcome of some experiment. For example, let's say in this one, you forgot to make sure that the plants were of the same starting height. So that would be a confounding variable. Ralph has an experiment. He wants to test the chemotaxis of Drosophila melanogaster. And so he puts one fly in this choice chamber here. On one end, he's going to put water. And on the other end, he's going to put spaghetti. He does this experiment one time. Now, this is obviously a terrible experiment. We need to help Ralph. So you can pause the video right here and think of ways to reduce the variance of this experiment. A big problem here that we can immediately see is that his sample size is tiny. He puts one fly in this. So normally when we do this kind of thing, we want to put maybe 100 flies or even more so that we can see how many of them go to one side and versus how many of them go to the other. And another method would be to increase the number of trials. So instead of just doing this one time, we could do this 10 times. That would give us a more accurate result because we could average all those times and reduce the variability. Last thing we could do is to eliminate some confounding variables. So what are some possible confounding variables in this one? Maybe the lighting in the lab is really weird, and maybe the lighting on one side is greater than the other, and somehow that has an effect on the fly. We don't know, but this is a really terrible experiment, and there are many, many ways to improve this. So what have we learned today? First of all, experiments allow us to see the effects of a particular change. And that's why they're very powerful. Hypotheses should be concise and testable, and they usually outline a correlational relationship. Your results should be both accurate and precise. It shouldn't be spread out, and it should be close to what reality is. And in experiments, we usually have independent, dependent, and controlled variables. So independent variables are the things that you change, and dependent variables are the things that change based on what you change. Controlled variables are the variables that you make sure stay the same. In an experiment, we'll usually have both control and experimental groups. Control groups serve as a baseline, and experimental groups are the things that you do things to. Negative controls are groups without the treatment done to the experimental group. So basically you could think of them as a default. Positive controls are groups that are given treatment that we know has an effect on it. Both of these types of controls, they serve as a baseline for us to measure against to see what sort of effect the experimental group has. When we do experiments, we want to have the variance as small as possible. And so some methods to reduce the variance include having a larger sample size, performing more trials, and eliminating confounding variables. So you want to get rid of all those variables that could be messing up your data. That was experiment design, and I hope that was helpful.